VGAS, the Coverage Navigation Report. Once the coverage view has been created, we can generate a navigation report. Here we have three groups of samples from different HRV projects using the HXB2 reference strain, and we're going to generate a genome-wide navigation report for the coverage view, and we have a 10% cutoff. So if we hit the update button, we generate the coverage view, and then we hit the navigation report button, we generate the navigation report. So I'm going to describe each tab. Nucleotide base counts, base position, the reference base of the reference we've used, which is HXB2, and with information for the three groups displayed after one another. So we have group 1, group 2 and group 3, and the thick line splits each group. Along the top we have ACGT. The question mark represents the ambiguous code, deletions, unsequenced count and unsequenced percentage. And the number at the top represents the number of bases for the entire group. So in group 1 we have 3,466 A's. And at position 2,490, group 1 has 152 A's, group 2 has 151 A's. We can also sort by clicking on the column header, and we can hide and display any of the columns as desired. So by clicking on the check boxes at the bottom, so if we're just interested in A's, we can see the A's. And double clicking on any row will navigate to that position as long as the navigation report is generated against the currently selected feature. So if we click on the GAG protein and we now try to navigate, we get a warning box. So the navigation report was generated against the genome wide with a cutoff of 10, but we are currently in GAG, so we can't navigate. So if we go back to genome wide, we can now navigate again. And this feature works throughout the navigation report. The second tab, single nucleotide variants or single nucleotide polymorphisms. Again, we've got the base position and the reference, and then any SNP which differs from the reference. So at position 1068 for group 1 we have a T where the reference was a G. And an asterisk next to the base denotes the most common base. It could be that within a group there is more than one SNP at a single position, and you can see this by the colouring of the rows. So at position 1095 we have two blue rows next to each other where the reference is a G, and for group 3 we have a T at that position and a G, and the T has an asterisk, so that denotes that this is the most common base. And the last three columns show whether the SNP occurs within a gene, a coding sequence, or another feature type. And if it occurs in more than one, then they're comma separated. So we can see that this SNP occurs within both Pol and GAG. Insertions. There are currently no insertions listed here because the sequences were imported from an aligned sequence FASTA file. The insertion would just show the base and the count in bracket. Deletions. Okay, group 1 has 22 deletions at position 6657. DNA group differences. Listed are all the positions where nucleotide base differences exist between any of the groups. However, I'll explain in the next example when selecting a protein. Protein group differences. There are no protein differences because we are currently viewing genome-wide, and a genome feature must be selected for these to be generated. You can open as many nav reports at once as you like for any gene or feature, and with different groupings, or with the same grouping but with different cutoff values. Okay, so we're going to show another example where we have a protein coding sequence selected. So if we select GAG, generate the coverage view, and generate the navigation report. And we'll go back to the DNA group differences. Okay, at position 44, the reference is a G, but for the three groups, group 1 has a GA, group 2 and 3 have GAC. And we can see in group 1 that we have 28% Gs, 24% As, and also the percentages for the counts in group 2, or the bases. At the bottom we can decide whether to show the percentage counts, the base counts, the quality scores. This is the average quality score, however we've not loaded any quality scores because this is not next gen data, and a codon check. So for the codon check, if we look at position 77, we see an A, and here we get the amino acid differences. So we have 117 Ks. So the amino acid K, the codon AAG, and the larger base represents the position of the base within the codon. So the second base is the A. So AAG is a K, AAT where we have 1 is an N, MAK1 is an X, and we have another K where we have an AAA which just occurs twice. Anything with a green underline indicates that there's just one amino acid possibility. So for the T we've got TTA which is an L, or TTG which is an L. Anywhere where we've got more than one possible base is comma separated. So position 171 for group 2 we have TC, where the T we have 2 codon possibilities where the third nucleotide base is the T in the codon, so TGT, KGT. This makes a C or an X amino acid, or we have a C where we have TGC which again just makes a C. SNP ranking allows us to sort of rank the changes. So if we order by SNP ranking we can see the bigger changes. And anything in grey represents a synonymous change which doesn't alter the protein. So we can see this row at position 1092 is grey, and the possible amino acids are always an A. So it's a synonymous change. And the protein group differences is the same as the DNA group differences but for the proteins, so the amino acid changes. And the navigation report can be exported into an Excel spreadsheet by clicking the Excel button on the top right. And all information will be saved in different worksheets. Okay, one quick tip when generating a navigation report. From the menu option, the settings, navigation report display, we can select which options we want to generate information for. If you have a genome-wide coverage view for a very large reference genome, you may want to select the options you require. So for a large million-plus base genome, the first option, nucleotide base counts, would display a row of information for each base, i.e. millions. So in this instance you might want to unselect the nucleotide base counts and then we don't populate the nucleotide base counts tab.